Hi, my name is Oscar. I'm a postdoc in the Applied Bioinformatics group in the German Cancer Research Center. And I'm going to present today an AI tool that we have been working on to predict the primary site using spatial transcriptomics. So, as you probably know, cancers of unknown primary are a heterogeneous group of metastatic cancers that are poorly differentiated and whose primary site is not known at the time of diagnosis. So the definition of the primary cancer type is usually achieved by pathology assessment using both H&E slides and immunohistochemistry, although usually are inconclusive and only are able to detect the primary site on 25% of the cases. And even within this, the diagnosis uncertainty is quite high. This leads to lack of treatments and, unfortunately, poor survival overall. So in the past years has been a lot of tests using machine learning and AI to predict the primary site, using mainly genomics and methylation assays. We wanted to follow a completely different approach in which because H&E slides are already part of the clinical pipeline to define the the primary site, we thought that annotating these slides might give the pathologists already some hints for the diagnosis of the primary site. For this, we constructed a dataset composed of bulk RNA-seq samples. We don't use any spatial transcriptomics here. We just focus on bulk RNA-seq, both healthy primary and tumor primary. This is because we want to be 100% sure that the ground truth is what we expect, and we don't want any uncertainty here. So our main sources are GDC Data Portal and GTEx, and also external data sets that help us to add underrepresented classes. With this, we are able to target 28 primary sites, covering most of the organs and tissues of relevance for cancer biology. And as you know, pre-processing of the data and how you show the data to the AI model matters a lot. So we thought about constructing a bioimage using the expression of the gene signature of a gene signature, so we can use a convolutional model to classify the primaries. We started by selecting the genes that were available overall in spatial Visium technology. And then we built a non-directed graph using collect tree database of transcription factors and targets. We focused first on transcription factors with at least 2 targets. And this gave us a gene signature of 3,900 genes, which we augmented later, adding some external features from the literature, both tissue-specific and cancer type-specific, that were also part of the collect tree database. So this gave us a total of 4,225, which conveniently can be transformed into a 65 per 65 matrix. So to do that, we perform first the Kamada-Kawai algorithm to construct a layout. And then with these coordinates, we try to solve the optimal placement problem using linear programming, also known as the Hungarian algorithm. And we come up with a template that we can use individually per sample. So this template, the transcription factors here that share targets are supposed to be close together, spatially close together. And the targets of these transcription factors common share between a transcription factor or link somehow by other transcription factors also should be placed closely together. And this is why we now can exploit this also like coordinate spatial information within this matrix. And we use a convolutional model, which is a COMNEX deep learning model, to do the classification. And apart from that, we do some data augmentation. The most important one is the mixed-up augmentation with a little tweak that we have called dominant mixed-up augmentation. So as you know, in Visium per spot, we have around 7 to 10 cells. And in our case, we might have a mix of tumor and healthy cells in, for example, the boundary of the tumor. So we want to teach the model to be biased towards the cancer signature. And we do that using this mixed-up augmentation that we mix healthy tissue from bulk samples and primary tumor from our training bulk samples from different primaries. And we call it dominant because no matter the composition, we retain the primary from the tumor. We have a minimum of 40% of composition of tumor per sample. But even in this case, as I said, the primary label of the class is going to be the primary side of the tumor. So we are trying to bias the AI model to identify the tumor signature here. Apart from that, we also have random erasing to mimic random dropouts in the feature space, which give us a total of 85,000 samples per training epoch. These augmented samples are created on the fly each epoch, so these are continuously changing. The metrics looks pretty well. So we have an overall accuracy of 97.5 accuracy. Also, we have some specific primaries that perform relatively worse than the best ones. As an example of this, for example, we have rectum that is usually misidentified as colon. And also we have larynx that is misidentified as bronchus and lung and mouth and tongue. That is understandable because they are like closely together and they, in some cases, quite closely related. And also these classes that are misidentified are usually with a low number sample size. So that's also one of the problems we have with these small classes, but we don't think it's a huge problem overall. And now moving to spatial, once we have trained this model, what we are going to do is treat each spot as an individual sample. Then we will collect all these predictions, and then we will filter out analyzing the probability distributions across these samples, trying to also find structures on the tissue. We started directly for ground truth samples. This means spatial transcriptomics data from primary healthy and primary tumor, in which virtually we could align our predictions with the primary, with the true primary. So if we get a prediction for each spot and we count how many classes have been obtained in this case, and we look at the top one, this is what we get. The predictions align completely with the local tissue. Much more interesting, distant metastasis, we have a collection of spatial data. And also virtually, we are able to align the predictions, the top one predictions, with the distant primary. You see here some yellow labels. These are also local, the anatomical tissue, healthy tissue that we are also able to identify in some of the cases. In taking that into account, we, as I said, we virtually can identify like the primary in all of these cases. So one of these cases that we also identify the anatomical size for you to see the annotation. Here, the first class is pancreas. If you count, like I said, how many of these spots have been classified as pancreas. The second option is lymph nodes. That is the anatomical side from where the biopsy was taken. Then we have some spurious predictions here and there. You can see that we have some heterogeneity in the predictions. So that's why we also follow an aggregative approach in which we recluster the sample with a higher resolution. We obtain a larger number of clusters, and then per cluster, we average the predictions, and we compute Hellinger's distance between the probability distribution of the top class and the second class. If this distance is below a certain threshold, we are not certain about the prediction for this part of the tissue, and we flag it as uncertain. This helps us to clarify a lot the annotation, and as you are going to see, also discover some structures. Here's another example of colorectal cancer that has metastasized into the liver, and you can see clearly the boundary of the tumor on the left, and the healthy local tissue. And now moving on to cancers of unknown primary, thanks to our collaborators from the Josep Carreras Institute in Barcelona, we got 20 samples that we could test. And first thing we saw is that the uncertainty increased a lot in our predictions. We also see a higher heterogeneity in general. And also in some tissues, we see, in some samples, we have a really complex tissue structures, which makes it even harder to annotate. Thankfully, we have the annotations from the pathologists, so we can compare also how our predictions align with these annotations. Apart from that, we are still analyzing all this data, but at this moment, we have 3 samples that we have some primary site suggestion from the clinical team, and for these 3 cases, our predictions align perfectly with the primary that is believed to be the original one. Here's one of them, is predicted as pancreatic cancer. Here you can see one of the problems that we are dealing now. So this is annotated by the pathologist as having fibrotic tissue, but is flagged as pancreatic, which we believe is both bias on our training data set and because of the nature itself of pancreatic cancer. So we are trying now to do some retraining and fine-tuning of the model to try to solve these problems. So I will show you some additional samples. Here we have a quite complex structure in which we have healthy tissue, we have small patches of tumor infiltrated. We also have fibrosis and lymphocyte-enriched spots. Our predictions, again, are quite heterogeneous, but if we follow an aggregative approach, we are able to discover some of the structures that were also annotated by the pathologists. And the interesting thing in this case is that if you take a look at the spots that were annotated by the pathologists as tumor spots, they completely align with a prediction of colon cancer. So in this case, we believe that this group sample might be colorectal, coming from colorectal cancer. This is another example in which we are able to identify both the local anatomical site, which is the lung, and the tumor that we believe is pancreas. Again, we have a mixture of fibrotic tissue and tumor. So yeah, it's sometimes hard to assess if the prediction as pancreatic is because of this bias or is the true origin. But we are positive that we can annotate and at least give some extra information for the pathologist to give some info diagnostics. With this, I want to thank, of course, my team in Heidelberg. Special thanks to Benedict, Xiaohan, and Emma, and our collaborators from Josep Carreras Institute and Cancer Research Center in Salamanca as well. So thank you.
Wonderful. Thank you so much for the talk. Are there any questions? I will get started with one then. And it's actually just a clarification. So forgive me if it is a little bit basic, but like your first training, you only have done it with bulk RNA sequencing, right? And then the
Yeah.
same model you applied to every spot in your spatial.
Yeah, that's correct. So we thought that if we use only bulk, then because the spots are pseudo bulk, it could translate directly, and this is the case. Also, we tested it with more like classical machine learning models, and the thing is that they perform quite well in general with bulk data, but this translation from bulk to spatial is when they usually fail, and it's like they are unable to generalize. So that's why we build this such a complex system with the bioimage input. And
I see. But then, my question for you is, do you know what would happen if you just take all your spatial data, you bulkify them, put all together, and then run your model as a single sample? Are you going to get like a similar results and accuracy or not?
yeah, that's a good question. So the problem with that is that with bulk data, we have in general a lot of samples available with all the primary sites. With the spatial, probably get a collection as with the number with the same sample size will be quite difficult. And pseudo bulking is sometimes tricky. We are, for example, trying to do that for the fibrotic tissue, and it's not working 100%. So it's like pseudo bulking usually is a little bit tricky for this model. So might work, but I will not use only pseudo bulking from spatial. Maybe a mix of both. Yeah.
I see. And then maybe just like a quick follow-up. In your model currently, you're not using any spatial information, right? Or neither morphology. Is it something that you plan to do?
Yeah, so we are not using the image itself. The thing is, there have been some papers in these past years that they try just by using AI image analysis to predict the primary site, but in COOPS it doesn't work really well. So we don't think that it will add much information to the model. Also, what we want to try is segmentation techniques with using AI segmentation techniques to uncover the structures and then try to aggregate and focus on the different structures in the tissue. Yeah.
Wonderful. Thank you. Any question?
Maybe precisely with the image, maybe you could train a separate model to detect fibrosis and to distinguish it from pancreas, and then to overlay the image with the prediction of the small cells.
Yeah, that's a good idea. Yeah, thank you for the suggestion. Yeah, that might be easier to do than just predict directly the primary from the image, but to flag the structure of fibrotic tissue and then remove that part from the prediction analysis. Yeah.
Any other question? In that case, we can thank the speaker again.
Thank you.